So now in this video we should talk about the modes of transposing or the transposition ways through which the transposable elements can move from one side to another. So see, on the broader basis there are two major modes of moving of transposons. One of them is the non-replicative way and the another one being the replicative way. As the name suggests, in the non-replicative way, there is no doubling of the transposon, but in the replicative way, there is the doubling or the replication of the particular transposable element. So what happens basically? When I talk about the non-replicative way, so what happens is that if this is your transposable element, it will simply cut itself from here and will move to a new place and be fitted there. But when I talk about the replicative way, in the replicative way, this particular transposable element will remain here. It will generate one more copy of transposable element, means it will do the replication, and then this new copy will be inserted here. So you will have two copies of the transposable element now. So we can clearly say that through this replicative way, the overall genome size keeps on increasing, because whenever the transposition will occur, the genome size will increase because the replication will take place. So these are the two broad ways. One is through non-replication and one is through replication. Now there is the concept of retrotransposons. If I talk about the retrotransposons, they are also moving from the one place to another. But what do they do is, here they generate their RNA. This RNA moves and here it converts into double-stranded DNA through the process of reverse transcription and then gets integrated. So overall, if we see, we come to know that the genome has been duplicated. No doubt this element is duplicated, but the way of duplication was different. So still it will be considered under the replicative mode of transposition. So now we should talk about both of them in details. First, let us talk about the replicative transposition. So as simple as that, the replicative mode of transposition can take place within the same DNA molecule also, or between two different DNA molecules. Now if we look at the summary of how the replicative transposition takes place, here is the diagram. Suppose we take the case where the transposition has to take place between two different DNA molecules, one being DNA A and the another being DNA B. And suppose this orange color is the transposable element. So now what happens? In the general terms, when this has to be transferred to the new DNA, the both of the DNAs have to first integrate and they have to form A plus B co-integrate structure. Now the A plus B co-integrated structure will be formed in a way that this particular transposable element gets divided, and then later on this will cross over. You can just imagine it as a rubber band which is now crossing at that particular transposable element location, and later on they will again divide, and when they divide and they separate, this process is called as resolution, which is guided by the enzyme called resolvase. So here the duplication of the transposable element has taken place, and here the formation of A plus B co-integrate has also taken place. So this is the general condition where the the replicative transposition takes place between two different DNA molecules. Moving into the details of this structure, how does this happen? Let's zoom into it. So here were the two DNA elements, DNA A and DNA B. There was one copy of transposable element which was available in DNA A, that is orange colored. Now this transposable copy has to be inserted into the DNA B in the region which is pink colored, which is also called as the target sequence. Now how will this take place? So basically there is an enzyme here which works. The enzyme name is transposase. The transposase enzyme basically makes the cuts here and here. That is, it cleaves the DNA at these particular sites, and similarly here and here. Now after the cuts have been made, what happens is that in the way we can say that this part has to combine with this part only, means the left hand side will combine with left hand side, the right hand side combines with the right hand side. So this will combine here. Sorry, I just made the mistake here. Left hand side combines with right hand side, and right hand side combines with left hand side. So basically what happens here, when the breaks have been made, this particular strand of DNA, which is the right DNA, right hand side, this will combine with the left hand side, and this left hand side will then combine with the right hand side of the particular DNA. Since this is circular DNA, most probably it is of prokaryotes. After that, such kind of structure is formed wherein the nicks or these gaps have also been created. Now we know what will happen here. The DNA polymerase enzyme will come and will do what? Will fill these gaps in a way that there will be flanking direct repeats which will be generated, but they have not been shown here. So these yellow colors are the transposable elements. These are the target sequence, and then there are also flanking direct repeats here. Now this uncoils. This rubber band will uncoil. It will form this kind of structure, A plus B co-integrate. So here this is our transposable element. This is also the transposable element. This was the target sequence part 1, target sequence part 2. Now in the next step, in the sixth step, there will be the crossing over between the two transposable elements. After the crossing over has taken place, the two of them will try to separate from each other, and when they separate, the two will divide and the transposable element copy will be generated separately. So this new copy is flanked by the direct repeats also of the target sequence. These pink colors are only the FDRs, because now these FDRs have been filled in by the DNA polymerase. And as I talked about, that the resolution will take place by the help of resolvase enzyme. In the non-replicative transposition, which commonly occurs very simply, the particular transposon has to move from one place to another, and that will be simply done as we had talked about in the this diagram. The cut will be made in the target sequence. The transposable element will bind to that particular sequence, and these gaps will be filled by DNA polymerase, hence generating the flanking direct repeats, and in this way the transposable element will be inserted into the target gene. The third type is with the help of retrotransposons or aided by retrotransposons. So the retrotransposons will work in the way that, first of all, the transcription of that retrotransposon will take place, or that particular gene will take, the transcription will take place to generate the RNA. This RNA, with the help of reverse transcription, will convert into double-stranded DNA. The double-stranded DNA then further finally integrates into the target sequence. This particular cleaves are made by the transposase enzymes, where the particular double-stranded DNA will be integrated and the FDRs will be generated. So there is the old copy of retrotransposon and the new copy of retrotransposons, both of them present. So this is also the mode of replication only, and this can also be called as the replicative mode of transposition. Now we have to talk about the transposable elements. We have to talk about the case of bacteria, then the case of maize, Drosophila and humans. So first of all, let us talk about the transposable elements in the bacteria. So For one thing that is very common is that the type of transposons found in bacteria are DNA transposons. Now in the bacteria there are no RNA transposons found, means there are no retrotransposons found inside the bacteria. The transposable elements in the bacteria can be of two types, one of them being the simple transposable elements, the other of them being the complex transposable elements. The simple transposable elements are very simple and only contain one main gene. These are also called as the insertion sequences. So insertion sequences are the simple transposable elements. These transposable elements contain only one gene or two which code for the transposase enzyme. Why this enzyme is required? So that it can make the cuts and help this to move to another place. So only a bare minimum of gene is present, that is the gene which encodes the transposase enzyme. So one kind of transposable element which is found in bacteria are the simple transposable elements which are called as the insertion sequences. So this particular whole of this TE is IS. Now there are some another kind of elements also which are called as complex transposable elements. But before that, let us talk about one of the examples of insertion sequences. The insertion sequence 1 is of total length of 768 base pairs and it is commonly found in E. coli. So if you want to learn this, you can learn. Moving on further, let us talk about the composite transposons. Now composite transposons have a very particular feature, that is, this transposon will consist of two insertion sequences on either side. So when the two insertion sequences and between them the group of genes are present, this whole is called as the complex transposon, designated as TN. So there is a very particular example given here, that is TN10. This is a composite transposon found in bacteria. The total length of the transposon is 9300 base pairs. This consists of IS10L and IS10R, wherein L and R stands for left hand side and the right hand side. In between there is the whole group of genes present which are enclosed on either side by the insertion sequences, otherwise called as simple translation elements. Simple transposition elements. The group of genes which are present in the center consist of one very important gene which is very particular to TN10, that is tetracycline resistance gene. So the composite transposons will consist of the insertion sequences on either side, and in between they will consist of the group of genes, one of them being the tetracycline resistance gene which is present in the TN10. Then there are the third kind of transposons found in bacteria which are called as the non-composite transposons. Now non-composite transposons are those transposons which are although big but they do not have the insertion sequences on them. So if the particular transposon element is big and on the either side it does not consist of insertion sequence, then it is, if these are not present, then it is the non-composite transposon. Can you think of why there is a need of insertion sequences on either side? The answer is very simple. Right now only I told you when I was teaching you insertion sequences that these insertion sequences consist of only one main gene which codes for transposase enzyme. What this transposase enzyme does, it makes the whole of the transposon mobile by making the cleavage. So if the enzyme is not present, the cuts cannot be made. If cuts cannot be made, then the transposable element would not be free, and if it is not free, then it will not be moving from one place to another. So the transposase enzyme has to be definitely present. And because insertion sequences consist of the transposable enzyme, so these insertion sequences are necessary for the composite transposons. Because although the composite transposons may have the tetracycline resistance gene, but they do not have the gene which codes for the transposase enzyme. So this transposase enzyme comes from these insertion sequences. When we talk about non-composite transposons, they do not have these insertion sequences, but still they have certain genes which can code for transposase enzymes, and through that they can be mobile easily. The best example is TN3. The TN3 is that which consists of the genes which have the transposase enzyme also, which codes for resolvase also, even for beta lactamase. So this is what you can learn here. It has been discussed already. TN3 transposon, it carries the genes for transposase, carries the genes for resolvase also, and it also carries the genes for the beta lactamase, and beta lactamase provides the ampicillin resistance. The last important one is the transposing bacteriophage, which is represented by Mu. There are some things which you need to know about them. First of all, these transposing bacteriophages can insert themselves in the host DNA during their lysogenic cycle, means their infection cycle. Secondly, they do not have the terminal inverted repeats. In the very beginning of the videos, when I talked about the transposable elements, I talked that not all of the transposable elements have TIRs. So in the mu, the TIRs are absent. It is very important to know. And this is particularly in the bacteriophage transposable elements. The whole of it is of 38,000 base pairs. There are flanking direct repeats which have to be compulsorily present. Then there are head and tail genes which code for head and tail of bacteriophage, and also then there are the other phage genes. So this is all about the transposable elements in bacteria. In the next class, we'll talk about the transposable elements in maize.